Let's go ahead and you can see my slides okay?
Yes.
All right, wonderful. So thank you for inviting me and letting me speak today. I'm going to talk to you about neutrophil heterogeneity, a pro-regenerative neutrophil that stimulates axon regeneration after central nervous system trauma. So, and then one more question. Can you see my mouse?
Yeah, we can see it.
Great. All right. So normally in a traumatic CNS injury, such as traumatic brain injury or traumatic spinal cord injury, when there's neuronal trauma, there is a release of neuronal intracellular proteins that are released into the extracellular space. These proteins bind to astrocytes, microglia, and to endothelial cells and antigen-presenting cells. And this creates a pro-inflammatory response, activating the inflammasome, leading to this recruitment of peripheral monocytes and neutrophils that migrate into the area. Generally, we think of this inflammatory response as detrimental to neuronal recovery and thought to, through the production of TNF-alpha, interleukin-1, and reactive oxygen species, actually lead to secondary damage to neurons after the initial trauma. However, there is a unique model that I work with called the optic nerve crush model, in which sterile inflammation actually promotes axon regeneration after the trauma. And this is completely opposite of all the other expectations and dogma that we see with neuronal trauma. And in this model, this is a very unique model. You make a microdissection to expose the optic nerve. You use a mechanical crush to transect the axons of the optic nerve. We then take a very small microsyringe and inject Zymosine intravitreally into the eye. The presence of Zymosine, which is a fungal wall extract, recruits large numbers of myeloid cells, neutrophils, and monocytes into the vitreal fluid space. These cells interact with retinal ganglion cells and then promote axon regeneration over time. And this is, as I said, unique. And this effect of Zymosine-induced sterile inflammation that promotes axon regeneration is specific to Zymosine. It's not non-specific to other sterile inflammatory proteins such as LPS. And so this is from a previous publication that came out of Dr. Segal's lab. I will orient you guys a little bit here. These are longitudinal sections of optic nerves that have been crushed. The eyeball and the retinal ganglion cells would be here. The star denotes where the crush injury is. The staining is GAP43. GAP43 is a growth-associated protein that is seen in regenerating axons. And then what you can see is that you get this nice linear staining pattern of GAP43 in the mice that underwent optic nerve crush and then were injected intraocularly with Zymosine. However, if you inject LPS or PBS intraocularly, you don't get that regeneration. You can take a grid where you can measure out the distance from the crush injury, and you can count the number of axons in a section, and then determine how much regeneration has occurred and how far this is from the crush site. And this is denoted down here in the bottom left-hand panel. And so when I first joined Dr. Segal's lab and then started to develop my own work, this is what we knew, but we didn't know anything about the immune cells that were infiltrating into the eye. And so I began my work by looking at what types of cells responded to the Zymosine injected intraocularly and which cells were recruited into the eye. And what I found when we did flow cytometry on the intravitreal space was that immediately following the injection of Zymosine, there was a huge recruitment of neutrophils into the intravitreal space. They were by far the most prevalent cell. The next most common cell was monocytes. And then after that, we had dendritic cells and T cells and B cells over time. But given that they were the predominant cell and we think of them normally as detrimental, the first question I asked was, what role do neutrophils have after the Zymosine injection? Is it possible that they're slowing down recovery and that the monocytes are responsible, or is it that they have an effect themselves? And so the thing that everyone needs to know is that neutrophils conventionally chemotax into an inflammatory space using the chemokine receptor CXCR2. And so we developed an anti-sera against CXCR2 to inhibit neutrophil migration through that pathway into the area. And so we split our mice into groups. They either received the control sera or the neutralizing sera against CXCR2. These mice underwent the optic nerve crush. We injected the Zymosine. I then treated the mice IP with either the control sera or the anti-CXCR2 and followed them out to look at regeneration. After 2 weeks of treatment and looking at that, we harvested the optic nerves. And then we looked at the amount of regeneration that occurred. And so on your right, you can see representative, these are clarity images of nerves that were injected with cholera toxin tracing for the axons. But once again, here on the left would be where the eye would be. The star, in this case, the black star, denotes where the crush injury is. And then the white axons that are growing distally across your screen represent the regenerating axons after the trauma. And if we do the crush and we inject PBS, we don't get much regeneration. If we use Zymosine, we get some quite robust regeneration. But what was really shocking was that when we injected the Zymosine and treated the mice with the anti-CXCR2, we had far better regeneration all the way to the optic chiasm, much further than we did with the Zymosine alone. And that was very interesting. So in addition to that, we were also able to look at neuronal viability. We take retinas, we do whole mounts and stain them for BRN3A and beta tubulin. And we would then count the number of RGCs that lived after the crush injury at the 2-week time point. And this is a naive retina for perspective of what a naive stain looks like. And if you do the crush injury, we lose about 90% of the RGCs. But with the Zymosine, we have preservation of RGCs, so we have preserved viability. And with the anti-CXCR2, once again, we have significantly more preserved viability than we do if we just crush the injury alone or just inject the Zymosine alone. And so the question was, well, did I block neutrophils? And so we once again did another time course where we harvested the vitreal fluid at serial time points throughout the crush injury after the treatment. And we looked at the relative proportion of the different cell types that were recruited into the eye. And what we found in the Zymosine plus the control sera is that, once again, neutrophils are still the first and the most predominant cell that infiltrates into the eye. But in our bottom panel, when I looked at the mice that were treated with the Zymosine and the anti-CXCR2, we found that we didn't get that early infiltration. In fact, we had a delayed infiltration, but neutrophils still found their way into the eye, and they peaked at a later time point around day 5 and continued to be present for the first week. Not only did neutrophils still find a way to get in there, and I'll get into more of the detailed flow in a few minutes, but when we looked at these cells, so if I did FACS sorting, flow cytometry sorting, and then did cytospins to look at these neutrophils on slides, what we found was that the neutrophils that were in the eyes of mice injected with Zymosine had a classic neutrophil morphology. They were hypersegmented, they had presence of granules, and they looked just as I would expect neutrophils to look. But in our flow sorted neutrophils from the mice that were injected with Zymosine plus the anti-CXCR2, we found that the neutrophils had this ring morphology, which is sometimes thought of as an immature morphology or an alternative morphology. They still had granules, so we were still able to visualize granules through a Giemsa Wright stain, but they morphologically looked different. We then did some more detailed flow cytometry to characterize these neutrophils and look at their expression of different neutrophil markers. And so we began with Ly6G. Ly6G is a classic neutrophil marker. And the red line is the histogram for the anti-CXCR2 mice or neutrophils, and the blue line is for the Zymosine with the control serum. And what we found was that the neutrophils in the mice that were treated with the anti-CXCR2 had lower Ly6G expression. They expressed CD14, which is a toll-like receptor 4 co-receptor, classically found on monocytes, not normally found on neutrophils. But our immature-looking neutrophils in the anti-CXCR2 treated mice had high expression of CD14. They had slightly lower expression of Ly6B, another neutrophil marker. And then they had, a large proportion of them were CD101 negative. CD101 is a neutrophil maturity marker. So CD101 positive cells are mature neutrophils, similar to Ly6G. And CD101 negative cells are immature neutrophils. We then took these sorted neutrophils and did some PCR to look at markers of alternative activation that is described in the literature, similar to the M1 versus M2 phenotype in monocytes. So arginase, IL-4 receptor alpha, and mannose receptor are all markers of alternative activation. And we found that the neutrophils that were present in the eyes of the mice that were treated with the anti-CXCR2 had higher levels of arginase 1, IL-4 receptor alpha, and then also mannose receptor, indicating an alternative activation state. We also did flow cytometry looking at our intracellular arginase staining. And so when we looked at Ly6G positive cells and arginase, we found that there was a higher percentage of neutrophils in the vitreal space and treated with the anti-CXCR2 that were arginase positive by protein level in addition to the transcript level. And one of the very common questions that we get when I first started to show this data, because they were showing these alternative classical M2 markers, are these monocytes? And so here I show you a representative plot in which we are focusing on these neutrophils, and these are our Ly6G negative population, but CD11b positive, so monocyte population. And when we look at the neutrophils versus the monocytes, we find that the monocytes are Ly6G negative as opposed to the neutrophils. The monocytes are MCSF receptor, CD115 receptor positive, whereas the neutrophils are not. And we find that the monocytes are not myeloperoxidase positive, but the neutrophils are. And so we feel very confident that we have sorted a unique neutrophil subset, not a unique monocyte subset. So I took these neutrophils and then we sorted them and purified them, and we sent them off for single cell RNA-seq. And here, what you're looking at is the UMAP plot of the single cell RNA-seq. And up here is the control treated group. And these are the neutrophils that came from the mice that were treated with the anti-CXCR2. And we had them split into 3 different populations that sorted out quite cleanly based on transcriptome. And they overlaid pretty nicely. And what you can see just visually looking at this UMAP plot is that population 1 is much higher in the controlled treated group, and population 3 is much higher in the anti-CXCR2 group. And when we look at this by individual markers looking at maturity or immaturity, the neutrophils found in clusters 1 of the anti-CXCR2 or in the NRS had high expression of mature neutrophil markers, and markers such as Ly6G, CXCR2, CEL or CD11b, S100A8, and CD0101. However, this population 3 that was more enriched in the anti-CXCR2 treated mice had higher expression of the immature markers that we see in neutrophils. So these are all transcription factors associated with immature neutrophils in their early stages. We also did the same thing where we looked at alternative activation markers versus classical activation markers in neutrophils, and found that our population 1 in both groups had high expression of classical activation markers such as TNF-alpha, interleukin-1, FAS, CCL3, CD24, whereas our population 3 that was enriched in our anti-CXCR2 treated group had high expression of alternative activation markers, markers such as arginase, TGF-beta, mannose receptor, insulin-like growth factor. And so from our single cell RNA-seq, we saw that there was heterogeneity in both treatment groups, but in the anti-CXCR2 treated group, there was an enriched population of immature alternatively activated neutrophils compared to the mice that received the control sera. So just to kind of delve into this a little bit deeper, you can take on any UMAP plot, you can take a gene of interest and overlay it onto your clusters and look at the relative expression of any individual gene within the clusters. And so in these examples here, the more red a gene is, the higher expression it is. And so if we look at those classical activation markers or classical maturity markers such as S100A8 or CXCR2, you see that cluster 1 has very high expression of these classical activation markers or classical maturity markers. Whereas those alternative activation markers such as arginase and mannose receptor, you find that the expression of those genes is almost exclusively found in that more immature population, which was very unique in when we separated out these populations. And so from the first part of my work, what we found was that in this model of immune-mediated axon regeneration, neutrophils are the predominant cell that infiltrate into the eye. You can block conventional neutrophil chemotaxis, resulting in a delayed infiltrate of neutrophils that enhances axon regeneration. And that enhanced axon regeneration is also associated with a unique neutrophil that was found, that Ly6G low, CD14 positive, CD101 negative. It had a ring nuclei and it was arginase positive. had a unique transcriptome that was differentiated from a classical mature neutrophil. So now we wanted to know more about these neutrophils. And so what we found was that it's very hard to take neutrophils out of an eye and work with them. There's not a lot of them. But you can take Zymosine and you can inject it intraperitoneally and you can get lots of neutrophils. And so if you inject Zymosine intraperitoneally and you wait 4 hours, you'll get a huge influx of mature Ly6G high neutrophils that migrate into the peritoneal space that you can lavage out. However, if you let that Zymosine sit, over a period of 2 to 3 days, an immature population of Ly6G low neutrophils will migrate into the eye or into the belly, and we can lavage those out. You can take these large numbers of neutrophils and pool them together and then use them to do things such as adoptive transfers or cell cultures and such. And that's what we did next. And so just to show you a little bit of the flow associated with these intraperitoneal neutrophils, they are similar to the intraocular neutrophils, in which when you wait 3 days, they have lower expression of Ly6G. They also have higher expression of CD14. When we do real-time PCR to quantify markers of alternative activation, we find that waiting the 3 days, you have higher expression of arginase 1, IL-4 receptor alpha, and monocyte receptor in those 3-day neutrophils compared to the 4-hour neutrophils. And finally, what we did was we purified them, we enriched them, and then we injected them into the eyes of mice that had undergone optic nerve crush. And so the optic nerve was crushed, and then instead of injecting Zymosine into the eye, we just injected the neutrophils. And so we either injected bone marrow neutrophils, those mature 4-hour neutrophils, or those immature 3-day neutrophils. And what we found when we looked at the number of axons that regenerated was that there were far more regenerating nerves and they grew further with the 3-day neutrophils than with any of the other neutrophil populations. So the next question is, a lot of people believe that, alternatively active monocytes have a very important role in all of this and that the neutrophils may be activating on the monocytes. And so to address the role of whether neutrophils function independently from monocytes, we used CCR2 knockout mice and wild-type mice. And you can take these intraperitoneal neutrophils and inject them intraocularly into CCR2 mice. And if you then look in the vitreal fluid, in a wild-type mice, you'll see the injected neutrophils. You'll also see this recruitment of monocytes. But in our CCR2 knockouts, you get the neutrophils, but you don't get the monocytes. And so now we have just the effect of the neutrophils in the eye instead of both the neutrophils and the monocytes. And what we found was that when we adoptively transfer these Ly6G low neutrophils into the CCR2 knockout, we got similar amounts of regeneration to if we injected them into the wild-type mice. And so here's some representative immunohistochemistry of those optic nerves, where the line denotes the crush injury, and the GAP43 staining shows you the regenerating nerves. And then we quantified that and showed that even in the absence of the recruitment of monocytes, the adoptive transfer of these Ly6G low neutrophils alone was sufficient in order to drive regeneration after the optic nerve crush trauma. And so in this part, I hopefully showed you some data indicating that Ly6G low alternatively activated neutrophils can be produced outside of the optic nerve crush model. We can harvest large numbers of them and adoptively transfer them into mice that have suffered optic nerve crush, and that these pro-regenerative neutrophils will stimulate regeneration independent of monocytes during that period of time. And so then we wanted to know, how are these neutrophils working? What is the method in which they are interacting with the retinal ganglion cells and stimulating regeneration? And so to do this, we moved out of the mouse model of crush trauma and into in vitro systems. And so you can take mice and you can take retinal ganglion cells out of the eye and purify them and put them into culture. And then I can inject Zymosine intraperitoneally and purify those Ly6G ring neutrophils out of those mice. And then I can co-culture them with those retinal ganglion cells and look at how much they stimulate regeneration. And this is what that looks like when we do this. And so if I just take retinal ganglion cells and I plate them, this is the cell body. You get a little tiny neurite that grows, and you can quantify the measurement of how long that neurite is. If I add Zymosine alone, I don't get any significant neurite outgrowth on its own. If I add CNTF, ciliary neurotrophic factor, which is a growth factor specific for retinal ganglion cells, you get these very long neurites that grow, and this nice, beautiful growth cone here. And then if I add these Ly6G low neutrophils, I also get this very long neurite that grows. And we can quantify this, and we can look at how many neurons go. And this is the average of 3 different experiments looking at hundreds of neurons each time. So the next question was, do these neutrophils directly interact with the retinal ganglion cells, or do they secrete some sort of factor that is soluble into the media? And so what I did to look at that question was we took the neutrophils, we put them into the media, we let them secrete factors for 24 hours. We then spun down the media, took the supernatant media off, but left the cells behind, and added just this neutrophil conditioned media into the retinal ganglion cells. We also took the media and we heat shocked it to 100 degrees Celsius and then looked at that effect and found that the conditioned media was able, in the absence of the neutrophils, was able to stimulate the amount of RGC neurite outgrowth similar to the neutrophils themselves. And if you heat shock the media, you abrogate that effect. So we took the neutrophils and I did a growth factor array and found that these neutrophils looked at the media and found that this media contained high levels of NGF and IGF-1, also TGF-beta. There were other growth factors that were also present in the media. I then did PCR, looking at the transcript level in those Ly6G low 3-day neutrophils compared to the 4-hour neutrophils, and found that NGF and IGF-1 was highly expressed by these neutrophils. I also did ELISAs quantifying the amount of NGF and IGF-1 that was found in that supernatant, and found that these neutrophils were producing growth factors. I then went back to my optic nerve crush model, and we looked at the growth factors in the vitreal fluid of those mice. And so if the mice underwent optic nerve crush, but then were injected with PBS, they did not have much expression of growth factors. But if they underwent optic nerve crush and then were injected with Zymosine, you had this increase in the presence of NGF and IGF. And when you use the anti-CXCR2 to inhibit the conventional chemotaxis and get those immature alternatively activated neutrophils, you got even higher levels of these growth factors. And just because you have a growth factor doesn't mean that it is responsible for the neurite outgrowth. So we then started to do inhibition assays to block those growth factors using an anti-IGF-1 antibody or an anti-NGF antibody, and looked at the effect of the neurite outgrowth from the neutrophil conditioned media. And so we would plate these RGCs and then produce that neutrophil conditioned media with all those growth factors, and then add in the neutralizing antibodies, and then co-culture that media with the RGCs. And what you can see is that if you inhibit either IGF-1 or NGF alone, you get a significant decrease compared to an isotype antibody or no antibody. But when you block both of them, you significantly abrogate the ability of these RGCs to regenerate neurites, similar to no treatment at all. And so then we moved on to the mouse model, the optic nerve crush model, looking at inhibiting these growth factors. And so once again, we injected Zymosine intraperitoneally. We waited 3 days. We got these Ly6G low neutrophils. We then added our neutralizing antibody or isotype control antibodies, and then injected both the neutrophils and the antibody into the vitreal fluid, and then wanted to look at the amount of axon regeneration. And what we found was that here are once again, some longitudinal sections of those optic nerves where the eye would have been here. The line denotes the crush injury. And this GAP43 staining shows regenerating neurons or axons past the crush site. Is that if you inject the neutrophils with the control antibody, you still get nice regeneration. That's quantified over here. But if you inhibit either the IGF-1 or the NGF or both factors, you lose the amount of regeneration you get. And it has a synergistic effect on those RGCs. And so from this part of my work, we determined that the mechanism of neutrophil-mediated axon regeneration is at least partially mediated by the secretion of growth factors, including NGF and IGF-1. I showed you that there are other factors involved, and we believe that other factors also have a critical role in maintaining RGC viability and axon regeneration. These two factors can stimulate it, and when we inhibit them, we prevent the regeneration. So the final thing that I want to talk about very briefly is the transition from mice to humans. Our human neutrophils capable of promoting axon regeneration. And so we started to explore this option, and this is an ongoing aspect of my project, in which we took HL60 cells, which are an acute myeloid leukemia cell line. So they're an immature myeloid cell type. You can culture them with retinoic acid, and they can be differentiated into an immature neutrophil in culture. And we also use some DG75 cells, which is a B-cell lymphoma cell, so also an immature cell type. And we started looking for growth factors that were being produced by these cells. And we found that these HL60 cells were able to produce NGF. The DG75 cells did not produce any growth factors that were associated with axon regeneration. We also looked at PCR. We looked at our markers of alternative activation. We found that these HL60 cells that we differentiated into immature neutrophils expressed high levels of arginase. And they also, not only did we find NGF in the media, but we also found it by transcript. And so we then took these HL60s and these DG75 cells and we cultured them with the RGCs. And we found that if you add these immature neutrophils into RGC culture, we get regeneration compared to the RGCs alone. This HL60 conditioned media also stimulates regeneration, but the DG75 cells do not. Because we've detected the presence of NGF and we knew that NGF stimulated neurite outgrowth, we then used an anti-human NGF neutralizing antibody and found that we could partially decrease the amount of NGF and decrease the amount of regeneration that occurred. If we took that human neutrophil conditioned media and heat shocked it, we could completely block the effect. And the other thing that we did along those lines was that we took those HL60 cells and those DG75 cells and those 3-day Zymosinin-induced mouse neutrophils, and we adaptively transferred them all into SCID mice. And we found that HL60 cells were able to stimulate axon regeneration after optic nerve crush, similar to those 3-day immature Zymosinin-induced neutrophils, but the DG75 B cells were not able to stimulate regeneration. And we were able to quantify that these have similar performance, indicating that at least, although this is a human tumor cell line, a myeloid leukemia, that the human neutrophil immature cell types do have the capacity to both produce growth factors and stimulate axon regeneration. And so this is a very shortened version of my paper. I hope this makes you interested in reading the whole paper. I'm happy to answer questions. But I hope that I showed you that there is a heterogeneity of neutrophils, and at least in certain circumstances, there's a unique subset of these neutrophils that are pro-regenerative, that have an alternatively activated immature phenotype that is characterized by being Ly6G low, CD14 positive, CD101 negative, arginase positive, and mannose receptor positive. These neutrophils can stimulate axon regeneration after acute traumatic injury, and that the mechanism in which these neutrophils function is both through interacting some with glial cells and through the production of growth factors that directly stimulates axons. So what I'm doing moving forward is, although this neutrophil story is partially uncovered, we're looking at how neutrophils interact with monocytes and what role they have and how do they work synergistically in stimulating immune-mediated axon regeneration and recovery. I'm looking at the chemotaxis of these immature neutrophils. How do they get into the area? If I block anti-CXCR2, they're chemotaxing through an alternative pathway that I have not identified yet. And then finally, I have some preliminary data looking at human neutrophils, at least in a cell line. And so we're now moving into getting human patients and getting blood to see if we can identify a human correlate similar to this mouse model. So I'd like to thank you guys for inviting me and listening to my talk, taking an interest in this. I'd like to acknowledge my mentor, Dr. Segal. He's the chair of neurology here at Ohio State. I'd like to thank my technician, Alex, who does a lot of the IHC and work, and then all of my collaborators and my funding for this. So happy to answer any questions you have. Thank you.
Thank you, Andrew, for this very elegant study. I think we could squeeze in a few questions. Maybe I can just start with asking, so you ended with the human correlates, right? I was wondering, if as you know from the clinic, right, there is this left shift phenomenon that happens very frequently under any kind of inflammation. Do you think that those left shifted immature neutrophils would already have the regenerative capacities, or is there something about, crossing the blood-eye barrier, or, you know, at what point do they acquire the ability to secrete those factors?
Yeah, so that is a very good question. And so, yeah, it is just kind of like follow up on that. It's very well known that in all types of infections and inflammatory situations, you do get that immature phenotype. So I can take patients who have had stroke or infection and find those ring neutrophils. But what's interesting about these neutrophils is I've done RNAseq and a lot of work with them in circulation. So I inject the Zymosine intraocularly and then looked at what the cells look like in the periphery. And although they have the ring morphology, they don't express any of that alternative activation. So they're not arginase positive, they're not mannose receptor positive, they don't transcribe NGF or IGF-1, I can't find TGF-beta on them. And so it's something about once they cross into the eye and interact with that zymosin. And so I don't know if there is a factor, if they're interacting with the zymosin directly and being activated that way, or if there is an antigen presenting cell or another factor that's being secreted locally at the site of injury that is responsible for that transition to an alternative activated state.
Okay, thanks. There was also a question or an attempt to link it to the previous talk. So because of this obvious, you know, link between zymosin and you know, both fungal components. Mm-hmm. So you also did transcriptomics. Did you notice the same signatures as Yanis did? So, you know, this oxidative phosphorylation or the ROS, did you pick up any of those?
So I haven't looked at any of that. So that is a great question. The metabolomics of neutrophil and myeloid cells is a next interest, but I did not go down the metabolic pathways in that same way. So it's on the list now. I did find that interesting.
Okay, thank you. Then we had a question about whether CXCR2 signaling is known to be linked to neutrophil maturation.
It is not. So neutrophils, mature neutrophils express CXCR2. It is a classical marker of their maturity, but it's not responsible for their maturity.
Okay, thank you. So then there was a question: are these pro-regenerative neutrophils found in EAE and other neurodegenerative diseases? That is very early to be said. So actually, with regards to EAE, very early on, I actually did a poster where I presented at ACTRIMS about, 2016, 2017, where we looked in the late phase of adoptive transfer EAE that Dr. Segal uses. And in the late stage, there is a brief increase in the infiltrate of neutrophils, and they do, although they're not ring morphology, they are arginase positive and TGF-beta positive. And so no one picked up on that project going forward, but we did notice that in EAE, there is a late phase infiltrate of those alternatively activated neutrophils. I don't know about in neurodegenerative diseases such as Alzheimer's, though. Okay. There's a question about whether you find the growth factors in the plasma of patients suffering from fungal infections.
I don't know. I have not looked. Great question.
Next question is, what activates the expression of the growth factors? You find the growth factor expression at day 3. Knowing that neutrophils are short-lived, could they be dying and therefore secreting the growth factors?
That is a good question also that I don't have the answer to. Why they're doing that, I don't know. Yeah.
Yeah, so this is where actually, I mean, so Kim's work would be relevant, right? Because the second talk, I don't know if you were, I know you were delayed.
Yeah.
So his lab showed that the EGF was secreted by dying apoptotic neutrophils.
I caught the tail end of it. Yeah, unfortunately, I missed the beginning.
Yeah. So Eugenio, I'm going to go ahead and ask a few more questions. Obviously, people are free to leave whenever they want because we are now over time. But so the next question would be, do eye neutrophils have similar regeneration capacity in other body regions, or do they behave radically different?
So that is a good question. So we are exploring that still right now. So we are exploring the idea of using the Zymosine locally in the EAE mice in collaboration with Dr. Segal. We are exploring the idea. We know that we can take those neutrophils and adoptively transfer them into other models. So part of my paper that I did not show you was we did some spinal cord injury, and we can inject these neutrophils into the spinal cord injury and stimulate regeneration in the same manner. And so the neutrophils themselves, once they have this alternative phenotype, can be transferred into areas of injury and have the same effect. But whether or not you can inject Zymosine in situ and get the same response is difficult. In spinal cord injury, Phil Popovich, who's also here at Ohio State, tried injecting Zymosine directly into spinal cord and found that it had two effects depending on how far away the macrophages were from the area of the injury. And so it's a really interesting paper that he wrote back in 2015 about that.